We are in the laboratory of laser molecular spectroscopy of the Technical University of Łódź, and our laboratory consists of many laboratories using linear and nonlinear optics. We are involved in cancer and immunology research, and I will show our laboratories. We are in the laboratory of Raman imaging. Raman imaging system consists of microscope and lasers for excitations, free lasers of different wavelengths, and we have also Raman spectrometer and detector. A Raman imaging system has also atomic force microscopy and near-field scanning microscopy. Separately, we have another atomic force microscopy system that can measure topography, adhesion, and stiffness.
The nanomechanical properties of different samples, including biological samples, can be analyzed in our laboratory by using the atomic force microscope designed by PIC Instruments. As you can see, the system consists of the inverted microscope, optical microscope designed by Leica, and AFM scanner enabling measurements in a range of 100 by 100 micrometers in X and Y directions, and 500 micrometers in Z directions, with positioning resolution equal to 6 picometers in X and Y directions and 0.9 picometers in Z direction. Nanomechanical properties are analyzed, of course, by using the dedicated software, which enables to analyze parameters such as Young modulus, stiffness, and adhesion.
Data acquisition is performed by using VTEC Control Plus system, enabling cosmic ray removal, noise filtering, and baseline subtracting. Here we have video image, Raman image, and Raman spectra for normal human duct. And here we have video image, and Raman spectra for cancer human duct. Using this system, we can also perform information about localization of biochemical components. Using this system, we can also perform atomic force microscopy and scanning near-field optical microscopy analysis without sample transfer.
Now we are in our second laboratory of nonlinear optics, infrared imaging and Raman imaging. Here we have the Raman imaging system, another Raman imaging system, and infrared imaging consisting of microscope, detector, and interferometer.
The Raman spectra and Raman maps can be recorded in our laboratory also by using the confocal Raman microscope designed by Reni Shaw. As you can see, the system consists of two lasers with different excitation laser lines, 633 nanometers, the red line, and 532 nanometers, the green line. The system consists also of the monochromator with two gratings with 600 and 1800 lines per millimeters, the detector and the optical part. The system allows us to measure the Raman spectra and Raman maps in air, but also in liquids. We can analyze different samples, including biological samples.
Here we got the Fourier transform infrared system from Agilent Technologies. We've got the spectrometer, and inside we've got the interferometer. This part of the system is the microscope, FTR microscope, with the focal plane array CCD detector. And this system gives us opportunity to imaging the tablets, the cancer cells, or the cancer slides of the tissue. And here you can see the imaging from the system. As you can see, we can monitoring the composition of the tissue and find the molecules that is inside in the tissue sections.
Our third laboratory is laboratory of Raman spectroscopy. The system consists of conventional Raman spectrometer with cryostat and handheld Raman spectrometer for cancer research, for brain, breast, colon, stomach measurements. Additionally, we have the system for in vitro incubation single cell system for incubation with drugs and vaccines. The system consists of laminar chamber and incubator.
Here we have high-performance Raman systems. This system is operating with 3 different excitations, 522, 785 and 671 nanometers. It consists spectrometers and very high speed CCD detector. This system allows us, thanks for the hand probe, to collect very high and very fast single Raman spectra. We usually use the system to analyze the brain and breast cancers, and as you see on the system, on the computer, you can follow the Raman bands in the Raman spectra.
In our laboratory, we cultivate human cell lines obtained from various organs, such as colon, stomach, breast and brain, both for normal and cancerous ones. For these tasks, we use a laminar airflow chamber and an incubator that provides constant growth conditions for cells. In addition, we also have a microscope for observing cells and automatic cell counter. In our work, we focus on studying the effect of supplements, antioxidants, especially in the form of vitamins, such as vitamin E and vitamin C, as well as commercially available drugs. In our biological laboratory, we cultivate our cells. We also perform viability tests, staining and fluorescence staining to obtain more precise and unambiguous results. Due to the pandemic time, we also direct our attention to investigation of mRNA vaccines and their impact on individual cell functions.
We are in our fourth laboratory of femtosecond spectroscopy using nonlinear optics. The system, femtosecond system, consists of femtosecond laser emitting the pulses of 100 femtoseconds. The laser is pumped by the green line of the diode laser. The single pulse from this laser goes to the regenerative amplifier, where it is amplified. Of course, the regenerative amplifier is also pumped by another laser. Here we have this regenerative amplifier. The output from this amplifier is 4 watts. It means that the energy of the single pulse is 4 microjoules, and then the output is split into 2 beams, and the pulses go to the optical parametric devices. Here we have many nonlinear crystals where different kind of nonlinear effects are produced, including second harmonic generation, third harmonic generation, fourth harmonic generation, signal, idler, summation pulses and difference pulses. So this system separately provides the energy from 2 nanometers up to 10 microjoules. So we have the full range from ultraviolet up to infrared. And here the pulses are split into 2 beams, pump and probe. 2 beams goes to the sample. The sample is here. This is the flowing sample with using the flowing system, and then the excitation, the effect of that goes to the spectrometer and to different kind of detectors depending on the excitation beams.
We are in femtosecond laser lab. We have a transient absorption spectrometer here. The heart of this system is this femtosecond laser. This laser is pumped by second harmonic of this diode pumped laser. For the experiment, femtosecond pulses must be amplified in the amplifier. Amplifier is also pumped by another laser, Q-switch laser. We amplified femtosecond pulses about 1 million times. In the end, we obtain peak power, that is instantaneous power per single pulse, about 40 gigawatt. These amplified pulses are used to seed these two optical parametric amplifiers. And here, two laser beams are generated. The first beam is called pump beam and is used to initiate the photochemical processes. And the second beam is called probe beam, which is used to monitor the changes introduced by the first beam. During the experiment, we changed the delay between pump and probe pulses using this delay line, which is here. For a probe, we can also use white light continuum, which is generated on this sapphire plate here. So in the end, both beams, pump and probe, are overlapped in the sample position, which is here. Usually we measure sample in solution, so we have to circulate the liquid using this peristaltic pump. To measure transient absorption signal, we have to detect probe pulse intensity using our detection system. This is detection system, monochromator, and two photodetectors. First photodetector is for visible spectral range. This is PMT, photomultiplier, and the second detector is for infrared spectral range. This is array of detectors actually. And the results of this experiment are shown on this software. All this spectrometer is controlled by this home written in LabVIEW software.
Dobra.